Clearing the malignancy.
In this video, I'm going to be talking about Kiwi and its actinidin effects. So, actinidin in Kiwi is a protease, not a receptor or transcriptional inhibitor. So actinidin affects on the Cd27 Cd70 axis and can partially cleave these extracellular loops, reducing receptor ligand dwell time, increased shedding, functional uncoupling. Literally, the Cd70 is coupled to the Cd27 via like adhesion molecule machinery, but actinidin and Kiwi can cleave through those adhesion molecules or machinery, therefore uncoupling the Cd27 Cd70 axis. Because if that goes on too long, AP1 loss. Here's important: actinidin affects integrin, Talin, Kindlin binding. Now that Talin Kindlin binding has to do with inside-out signaling that cancer cells and tumors like to use for their processes and destabilizing the immune system. But actinidin in kiwis leads to reduced mechanical tension across integrins, less Talin unfolding, less Kindlin stabilization. Therefore, cancer cells and tumors cannot use the inside-out signaling mechanism. Okay, another really important thing about kiwis is it prevents the Talin integrin activation caused by calcium. Because calcium is important, but calcium can support the Talin, Kindlin, integrin inside-out signaling that could destabilize the immune system because cancer cells exploit that. But Kiwi can interact or can intervene by cleaving the adhesion molecules or the integrin and Talin itself. Therefore, integrins remain in low affinity. It also reduced immune synapse stability and cell migration persistence caused by Talin and integrin activation by calcium. But kiwis has to interact or intervene because calcium is needed to activate calcineurin, so that calcineurin removes phosphate groups from NFAT. Then NFAT translocates to the nucleus so that it can interact with AP1 to revive exhausted T cells with IL-2. So Kiwi allows this to happen by allowing the calcium to activate the calcineurin, but at the same time, Kiwi offsetting the negative effects of calcium because Kiwi cleaves the Talin Kindlin, therefore allowing the uncoupling of the Cd70 Cd27. But without Kiwi, the calcium will actually activate or enable the coupling of the integrins, Talin, Kindlin, and then let's say tunneling nanotubes, the Cd70. But Kiwi, like I said, will interfere with the whole Talin Kindlin binding. Actinidin in Kiwi also reduces NFKB that leads to AP1 loss, which makes it difficult to open up the chromatin. And it does this by blunting the NFKB triggers that are caused by Cd70 from tumors. And Kiwi does this via proteolysis, which is a process. The next one is a critical one. They are called Succinate and Fumarate. What they do is that it reduces immune activation, therefore reduced glycolytic pressure. Thanks to the actinidin from Kiwi acting on Succinate and Fumarate. Therefore, less glycolysis, which is critical because tumors feed on glycolysis. Also, actinidin reduces glutamine flux. Glutamine is a fuel source for tumors. Also, proteolysis, less macrophage HIF1 A. Stabilization due to less succinate accumulation. Also improved mitochondrial throughput due to reduced fumarate spillover. So mitochondria is extremely important for immune cells because they get exhausted. So thanks to kiwis, mitochondria is improved as well, leading to reduced succinate fumarate, less H3K9ME3 persistence, which is a repressive mark on the chromatin that will not allow the chromatin to open. Next one is critical. Actinidin inhibits exosome formation because actinidin in kiwis disrupts this at multiple levels because exosome biogenesis depends on membrane scaffolding precision. So actinidin cleaves tetraspanins CD9, CD63, and CD81. Therefore, cargo sorting becomes noisy. So basically, exosomes, they have cargo, such as mRNAs, miRNAs. What those cargoes do is that it prevents the trimming of the poly A tail on the Cd70 by adding caps, these caps at the ends of the poly A tail to prevent the trimming. Therefore, the poly A tail stays on the Cd70, and the Cd70 continues to oppress Cd27 on immune cells because Cd70 is on the tumor. Okay, without the exhausted T cells will not be able to be revived, or they will become exhausted because of ongoing NFKB, leading to AP1 loss because of the Cd27 Cd70 axis. Another critical one here is that reduced inflammatory signaling leading to reduced NS-MACE activity. So NS-MACE is a machinery that helps drive ceramide-driven ILV formation. So ILV is like the vesicles inside the MVB sacs that holds the cargo or attaches to the cargo. So actinidin leads to ceramide ESCRT coupling being weak. Cleaving of integrins by actinidin in kiwis leads to the vesicles not being able to guide the exosomes. So as a result of these vesicles becoming mistargeted, the exosome quantity goes down, which is critical. Less inflammatory cargo, because exosomes, they hold cargo that can wreak a lot of havoc. Also less metastasis, upregulating biologically quiet vesicles. Okay, so quick summary: Cd27 Cd70, proteolytic dampening thanks to kiwis. Kiwis uncouples integrin, talin, kindlin, which is inside-out signaling that cancer cells and tumors use to destabilize the immune system. So kiwis weakens that by doing the uncoupling. Kiwis also reduce NFKB, AP1, that leads to the collapse of AP1, aborting the activation of exhausted T cells with the IL-2 that they need. Kiwis prevent that by reducing NFKB, which leads to the AP1 loss. Kiwi also reduces succinate fumarate, which is what aids cancer, therefore allowing epigenetic recovery at the chromatin level. Okay. Also, kiwi reduces exosomes by disrupting the tetraspanins, therefore less vesicles that are holding on to the exosomes. And a bonus here is that so actinidin shortens signal half-life, or the signals that cancer uses to destabilize immunity is shortened. That's critical, especially at the end of a fast, because immune system can be deactivated for days, because it takes them days to reactivate, while tumors, they only have 5 minutes to 30 minutes to hog up glucose or protein as a fuel source. And actinidin might shorten that. So like, comment, share, subscribe, and I'll see you in the next video.